All right. Hello, everybody. I'm Kushal. I'm in Miss Ahmed's B6 biology class, and my group's project for the STEM SOS 2018 project was to construct a cell in general, right? So let's just say an animal cell, all right? First, let's start off with our background research. Our main idea was to construct an animal cell, or any type of cell in general, because they pretty much have the same idea. Some keywords to describe our project are mitochondria, cytoplasm, and nucleus. These three are all very important organelles inside a cell. For example, the mitochondria is the powerhouse of the cell. It generates the power in a nutshell. The cytoplasm basically covers the entire cell and holds everything in place. Because if nothing is in place, then everything will just be floating around, and that's not good. Now, the nucleus, that's the brain of the cell. Now, without a brain, nothing can function, correct? I hope so. Some principles presented in our project are lactic acid fermentation and glycolysis. Lactic acid fermentation is where lactic acid, it can happen both in plant and animal cells, uh, takes place inside your muscles due to anaerobic respiration. Glycolysis is the step before this. It's where sugars break down in the cytoplasm, and it gives the cell two choices that it can overcome based on the presence of oxygen. If there is oxygen, it'll go through aerobic respiration, but if there's not, anaerobic. So now, our driving question for the project. Our driving question was what makes prokaryotic or bacterial cells and eukaryotic or animal cells different from, and why are they like that? Our hypothesis is that prokaryotic cells do not have a nucleus, and they're built that way because over time eukaryotic cells have evolved and they've adapted to their situations, and prokaryotic cells, they've just been lazy and they just said, nah, we're not going to do it. Our materials for this project were styrofoam balls, toothpicks, and other desktop items like glue and scissors. All right, so with this being said, our safety precautions weren't that high because they're all desktop items. I mean, instead of just intentionally sticking your hand in a bottle of toothpicks so you can poke your hand and putting gloves on so you won't get glue on your hands and being careful with scissors because you want to keep your hands. So now our experiment. We first began by modeling both of the cells, prokaryotic and eukaryotic, by looking at some references from the internet and observing their differences. I have to say that it was quite fascinating. Bacterial cells are so different, but also so similar to the eukaryotic cell structure, and it's just really mind-blowing. After modeling them, we looked to see if our hypothesis was correct, which it was. The safety for this project was very minimal, like I said before. We did not much have heavy-duty work to do. All we had to do was to wear gloves when we were handling glue and other such materials. Our procedure now is the good part. So first, we went out and bought all the items we needed, obviously, like glue, toothpicks, styrofoam balls, and labels. So first of all, we cut the biggest styrofoam ball in half so we could get a bird's eye view of the cell. Then we modeled our organelles based off of how they look like. So for example, the mitochondria had folded membranes all packed in together to one little powerhouse, right? And it's just laying there in the cytoplasm. The cytoplasm, we put like this white Play-Doh all over it, and we did that. And we just modeled it like that. So again, I'd like to thank you for listening. This is Kushal and Batipudi. My group members are Met and Honeywell, and I'm in Miss Ahmed's B6 biology class. Toodles.